Hi, my name is Juan Restrepo and this is my biology project. First of all, we need to understand what are the physical bases of inheritance, how were the principles of inheritance discovered, and how are the traits discovered, or how are the traits inherited. First of all, we have to understand what is inheritance based of. Inheritance is the way by the traits are inherited to the offspring, by meaning how are the traits passed to the next generation. The trait is something you have and it's always in your genes. It's always located in your genes. It will never be lost. The the traits are made up by a specific genetic structure. I will show you one that is made up of adenine, cytosine, thymine, and guanine. This genetic structure builds up an unit of inheritance that is called gene. A gene is a unit of heredity that encodes information needed to produce proteins, cells, and organisms. These locations of the genes are on a chromosome they are located in a chromosome and its specific place are the locus. Now, traits are located in alleles in chromosomes. I will show you chromosomes. For example, these are a pair of homologous chromosomes. At this part of the locus, the allele contains the same gene. At this part of the chromosome, the allele have different genes, but they are the same between both. But at last, these two genes, these locus, they are different. They got different genes. They got different traits. They got different alleles, so they are different. Now, the bases of inheritance were discovered by a priest called Gregor Mendel. He is considered the father of modern genetics. He discovered the Punnett squares, that they are a mathematical probability to discover which will be the traits inherited in an organism. Mendel did an experiment with two true breeding plants which contained a dominant allele with purple color of petals and a recessive allele with white color of petals. Gregor Mendel for this experiment chose these two true breeding plants that got their traits inherited by generations by self-fertilization. So he cross-fertilized these both plants. So now we got to do the monohybrid cross that Mendel did with its two true breeding plants, which were, remember, the purple is dominant and the white is recessive. As we can see in here, we got phenotype, purple dominant and white recessive. Always, it's characterized in the Punnett squares by the dominant to be in uppercase and the recessive to be in lowercase. Now, in the F1 progeny, that will be the result between the cross of the parental generation of these two different plants. We always in the F1 progeny get an heterozygous result, as shown in this square. We can see there is the four results are heterozygous. Each square in the monohybrid cross represents the 25% of probabilities. So if one square of that represents the 25%, by for example, if you get an homozygous dominant in one square, you got the 25% of probabilities. The phenotype demonstration will be dominant of that characteristics. So in the genotype, we put the 100% or the 4 squares in ratio or fractions, and the phenotype, as the heterozygous got the dominant allele 2 and the dominant is, well, have a dominance over the recessive, the phenotype will be shown as flowers with the purple color of petals. Now Mendel he was strange with the results, so he did a second cross between two 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 offsprings of the F1 progeny. So he got another results, which are the F2 generation. For the F2 generation Punnett squares, you must use the results obtained in the square you made before. So all of them are heterozygous, so you made an heterozygous Punnett square shown here. As you can see, you put the ovum donor and the pollen donor as heterozygous, so you make the cross P with P. They are both uppercase, so they go uppercase. P uppercase with P lowercase is heterozygous, and these two make an homozygous recessive. So in the genotype, we will get 2 squares with an heterozygous result. That is the 50%. Remember, 1 square represents the 25%, and there are 2 of 4, so that's the middle. You got one homozygous dominant square and one homozygous recessive square. In the phenotype, that doesn't change too much, because you got the 75% of probabilities for the flower petal of color to be purple, as you got 2 heterozygous over dominant over recessive, and you got 1 homozygous dominant, and you got only 1 square with an homozygous recessive result. That is the 25% against a 75%. Now we gotta make the hybrid cross. What is the hybrid cross? It's a Punnett square, but with 2 traits, not 1 but 2. So the square must be more extensive. As there are 2 traits and there are 4 squares by trait, we multiply these two quantities, 4 by 4. That is 16. So the Punnett square must have 16 spaces to blank out. We gotta make with seed shape and seed color. As you know, the seed shape, you got two alleles, a dominant and a recessive. The dominant is a smooth, and the recessive is a wrinkled shape. For the seed color, we got a dominant color for yellow and recessive for green, as shown here. We got the seed shape and the seed color, the dominant and the recessive. We will represent S in uppercase and in lowercase for seed shape, and Y in uppercase and lowercase for the seed color. We didn't make the F1 square by a single reason, that is that always the results in the F1 progeny are heterozygous. So we ain't doing the math, that is wasting time. What we interest is the Punnett square for the probabilities in the matching in the mating of 2 offsprings of the F1 progeny. So we get this square of here. As you can see, you got to do the same process you did with the monohybrid cross. This is a lot more extensive, and now you got 2 traits. For example, you got a cross between 2 homozygous dominant, so you put two uppercase S and two uppercase Y. Why? Because there are two traits. Now, the genotype will be different. Why? Because there are different squares. It's a different math, so it's not 25% anymore, but the 6.25%. That is 1 divided 16 by the 100. So we get a percentage that is the 6.25%. So we, in this Punnett square, we get 14 squares showing heterozygous results. That means the 87.5% of squares. This doesn't mean every squares got the same characteristics of heterozygous. There are some that got wrinkled seed shape or other that shows a smooth seed shapes, but they changed. They got a dominant allele and a recessive allele, but not in the same trait. We get one homozygous dominant and one homozygous recessive trait in the whole square. Only two squares represent homozygous results. The phenotype is different. Obviously, there should be more results with dominant traits, and that is shown in here because 9 squares of the Punnett square represents the 56.25% for a seed shape, for a smooth seed shape, and a yellow color of the seed. 3 of the squares, 3 of 16, got a wrinkled seed shape and a yellow color of seed, that is the 18.75%. As you can see there, you remember wrinkled is recessive. You got one recessive and one dominant. That is the meaning of being heterozygous. But in the next example, it will change because we got 2 squares, heterozygous 2, of a smooth seed shape and green seed color. That means the 18.75% of probabilities, and only one square, that means the 6.25% of probabilities, you got a seed shape wrinkled and a color seed, or a color of the seed green in this type of plant. What we want to show you is that first, for being sure, the results in the genotype and the phenotype, the percents summed up must get the 100%. If not, you gotta repeat all the process. We can use the Punnett squares for 3 traits, but that will take more longer because remember, 4 by 4, 16 by 4, other 4 is 64 squares to do. So we're leaving until the F2 progeny of the D Harvey cross. Thank you very much. This is my project.